Bismillahirrahmanirrahim. Assalamu alaikum, dear student. The next topic we are going to start higher animal taxonomy. Traditionally, the Animalia have been considered monophyletic. We already know about the monophyletic when we consider that all organisms are originated from a single ancestor. Then we use a term that is called the monophyletic. When we consider that all animals have separate ancestors, then the term is used that is called the polyphyletic. So traditionally the Animalia have been considered monophyletic, having single ancestry because of impressive similarities in the cellular organization. About 0.6 billion years ago, at the beginning of the Cambrian period, an evolutionary explosion occurred that resulted in the origin of all modern phyla. During the Cambrian period, rapid origin and diversification of animals occurred, and this rapid origin and diversification of the during the Cambrian period, evolutionary explosion occurred that resulted in origin of all modern phyla, uh, and some animals are now extinct from that phyla. This rapid origin and diversification of animals is called Cambrian explosion, but it occurred over a relatively brief 100 million years period. This rapid appearance of all animal phyla leads some scientists to believe that animals may be polyphyletic. In each group have a single separate ancestor in case of the polyphyletic view, because such a rapid divergence of many kinds of animal from a single ancestor seems unlikely. Taxonomic levels between kingdom and the phylum are used to represent hypotheses of relatedness between animal phyla. Many zoologists recognize three major groups within the animal kingdom. These groups are often called branches and include the Mesozoa, phylum Mesozoa, the Mesozones, phylum then Parazoa, the phylum Porifera, the sponges, and Eumetozoa, all other phyla. The Eumetozoa are further divided into two groups based on the body symmetry and embryological characteristics, as in the table 7.4. In this system, the bilateral symmetrical animals are divided into two large groups based on the similarities and differences in the early development of the animals. The study of comparative embryology is based on the observation that embryological events may be similar because of the shared ancestry. As with other comparative studies, however, embryologists must sort homologous development sequences from analogous developmental sequences. The table 7.4 here, traditional categories and that of the alternate categories. First of all is the phylum Mesozoa. Traditional category is the Mesozones, and Mesozoa are further divided into violet Echinodermata, hemichordate, and chordates. These are included in the Deuterostomia, and they have traditional category Deuterostomia, and same alternate categories also the Deuterostomia. And arthropods and nematodes are included in the Ecdysozoa, and Platyhelminthes, Nemertea, Mollusca, and Lida. These are also called the Bilateria, containing bilateral symmetry, and Protostomia, and alternate category they have Lophotrochozoa, and Cnidaria and Ctenophora, Eumetozoa, and Radiata, containing radial symmetry. Porifera are the Parazoa. Parazoa is a group of animals comprised of cells that do not work together in a coordinated manner and are not organized into tissues. Phylum Porifera, the sponges, and Eumetozoa. These are the animals that are multicellular, in which cells are organized into tissues, organs, and organ system. And now, students, we are discussing the major topic, the main important topic, the main topic that is called the Protostomes and the Deuterostomes. Differences between the Protostomes and the Deuterostome. This is a much important topic because of the examination point of view. Table 7.4. Two interpretations of higher taxonomy of selected animal phyla. First, in the first box, phylum. Second, in the traditional categories, and third one is the alternate category. First one is the Mesozone. Mesozones are the tiny, simple worm parasites that were once thought as of intermediate between the protozoa, motile single-celled organism, and the metazoa, multicellular organisms, multicellular animals. However, now they are considered as a degenerate metazoans, phylum Mesozoa, the Mesozones. And they have a traditional category Mesozoa, but alternate category, nothing is alternate category. And the second one is the Porifera, that includes the Parazoa, and next is the Cnidarians and the Ctenophorans, Eumetozoa and Radiata. Parazoa, Eumetozoa and Radiata, these are the diploblast consisting of two layers, two body layers. And the in the phylum box, next one is the Platyhelminthians and the Nemertians. These are Bilateria containing the bilateral symmetry, and then the Protostomia, and then Protostomia, then Deuterostomia. Protostomia contain the Lophotrochozoans, Lophotrochozoa, and next is the molluscans, annelidans, and arthropods and nematodes. These all are included in the Protostomia, and arthropoda and nematode are also called the ecdysozoa because ecdysis occurs in these. And the next one is the echinoderma. Now the protostomes and the deuterostomes, these are the important topics. Protostomes are what? We know the development process. Development starts from fertilization, then cleavage, then blastulation, then gastrulation, then organogenesis. In the blastulation, blastocele is formed, and then blastulation leads to the gastrulation. And in the gastrulation, mouth-like opening of the archenteron on the surface of an embryo in the gastrula stage is called the blastopore. And blastopore is an opening by which the cavity of gastrula communicate with the exterior is called the blastopore. If the blastopore changes into the mouth first, then these are the protostomes. Proto mean first and stome mean mouth. In case of the deuterostomes, if the blastopore changes into anus first and mouth is formed later, then we can call it the deuterostomes. तेरा काम आ चुका. The protostome and deuterostome. Blastopore is an opening in the cavity of archenteron, or it is the opening by which the cavity of gastrula communicate with the exterior is called the blastopore. The fate of blastopore here it is, if it is changed into the mouth, then the category is known as protostomes. The animals are known as protostomia. If the blastopores is changed into anus first, mouth is formed as a secondary opening, then we can say the deuterostomes, deuterostomia. So the protostomes have some differences with that of the deuterostomes. In the protostomes, first of all, differences. First of all, these include the phyla, animals in the phyla that are Platyhelminthes, nematodes, molluscans, annelid arthropods, and others. In these animal phyla, the protostomes, proto mean first and the stome mean mouth. Here the mouth is formed as a first opening and anus is formed as a second opening. Next is the pattern of cleavage. Cleavage is what? When a fertilized zygote is changed into a ball of cells is called the cleavage. Now the cleavage is also of two types, spiral cleavage and the radial cleavage. In case of the protostome, spiral cleavage is present. Here the mitotic spindles is oriented obliquely to the axis of the zygote. This orientation produces an 8-celled embryo in which the upper tire of the cell is twisted out of the line with the lower cells. Second characteristic that differentiate the protostome from the deuterostomes is the spiral cleavage. Here the mitotic spindle is oriented obliquely to the axis of the zygote. This orientation produces an 8-celled embryo in which the upper tire of the cell is twisted out of the line with the lower cells. In case of the spiral cleavage, The cells are not aligned over each. The next category is the Deuterostome. Include the animals that are present in the phylum Echinodermata, phylum Hemichordata, phylum Chordata, and others. In this case, the radial cleavage is present. Radial cleavage is what? When the mitotic spindle oriented perpendicular to the axis of the zygote and result in the embryonic cells directly over one another. When the cells are present over one another, this type of cleavage is called the radial cleavage. Cells are aligned directly over each other. The cleavage type is called the radial cleavage. In case of the cleavage, the next type of cleavage in case of the Deuterostomes is the indeterminate cleavage, meaning that the fate of the blastomeres is determined later in the development, and if the embryonic cells are separated, they can develop into the entire individuals. And next is the coelom formation. In case of the Deuterostomes, the manner of the gut tract and coelom formation differ from that of the protostome. In case of the Deuterostomes, coelom formation is called the interocoelous type of coelom formation because it is formed from the out pockets of the gut. While in case of the protostome, it is formed from the splitting of the mesoderm. Some protostome possesses a kidney-shaped larva, kidney bean-shaped larva that is called the dipleurular larva. There is, however, no single kind of deuterostome larva stage. Even though these higher taxonomy groupings are traditional, a number of phyla, mollusks, nematodes, and others do not fit well into either deuterostomes or the protostome groupings. Cladistic analysis and molecular approaches to taxonomy are resulting in alternate groupings and gaining special widespread acceptance. Deuterostomes. In case of the deuterostome, the blastopore changes into the anus first and the mouth forms later, and the cleavage is radial. When the mitotic spindles are parallel and perpendicular and the cells are aligned directly over each other, and the cleavage is also indeterminate when the cells can adjust at any time. The fate of the blastome is determined later in the development, and the type of the coelom formation is the interocoelous coelom formation. The coelom develops from the out pockets of the gut. Next is the, in case of deuterostome, dipleurular larva is present, kidney bean shaped larva. And next is the here in the animals belonging to phylum Echinodermata, Hemichordata, and Chordata are included in the deuterostomes. Now the table 7.12. Here protostomes and the deuterostomes, diagrammatic differences between the protostome and the deuterostomes. First of all, you can see the cleavage type spiral in case of the protostomes, and while in case of the deuterostomes it is type radial, and spindles are oriented after first and second cleavage at 45 angle to animal pole and the vegetal pole of the egg in case of the spiral cleavage. While in case of the radial cleavage, the mitotic spindles are parallel or perpendicular to each other. Second one category is the blastula is formed. In both cases, same blastula. In the third one, gastrula is formed. These are the developmental stages: zygote, fertilization, then cleavage, then blastulation, then gastrulation, then organogenesis. Gastrula. Here the blastopore is shown. Early mesodermal cells, and same is the case in case of deuterostome. Coelom develops from the out pockets of the gut. You can see in the diagram out pockets are appearing from the gut to form the coelom. In the next diagram, the blastopore changes into the mouth in case of the protostomes, while in case of the deuterostomes, anus develops in the region of the blastopore, and now the cells are formed. Now the layers are formed: ectoderm, mesoderm, and the endoderm, and gut is formed on the inner side of the embryo. Next is the, in case of the C, mesoderm split to form the coelom, while in case of the deuterostome coelom is formed from the out pockets of the gut. Both are similar in morphology. Next is the D. Tocopher larva is present, top-shaped larva is present in case of the protostome, while in case of the deuterostome, diplerula larva is present. But no single kind of deuterostome larva stage is present in case of the deuterostome, and diplerula larva is present in case of the deuterostome. Procophore larva is present in case of the protostomes. The further differences between the spiral cleavage and the radial cleavage. Here the zygote, then 2-cell stage, 4-cell stage, then 8-cell stage, and again the 8-cell stage. In the spiral cleavage, the mitotic spindle is oriented obliquely to animal vegetal axis of the zygote. This orientation produces an 8-celled embryo in which the upper tier, row, or the layer of cells is twisted out of the line with the lower cells. And in case of the radial cleavage, it is called because the embryonic cells are arranged in the radial symmetry around the animal axis. Animal vegetal axis, each cleavage furrow is oriented either parallel or perpendicular to the animal vegetal axis of the zygote. So the blastomeres are oriented directly over one another. Zygote, then the 2-cell stage, 4-cell stage, 8-cell stage, and next one is the 8-cell top view. This is all about the higher animal taxonomy. These are the key events during an animal development. First of all, gametes form, sperms and eggs form, and then they mature. Then fertilization of egg and sperm fuse. Then cleavage. Zygote subdivides, determinants partitioned in the blastomeres. Then gastrulation occurs and germ layers are formed, and after the gastrulation, organogenesis occurs. Body organs form, cells interact and differentiate, and after the organogenesis, growth occurs, organs increase in size, adult body form is attained. During the blastulation, two poles are observable in the ovum. First one is the animal pole, that is present at the point on the surface of an animal ovum nearest to the nucleus and normally at the opposite side of the egg from the aggregation of yolk droplets. And second one is the vegetal pole. More yolk area, vegetal pole is called the vegetal pole. Animal pole opposite to the vegetal pole, and it is nearest to the nucleus is the animal pole. Animal pole nearest to the nucleus and opposite to the vegetal pole, and vegetal pole is the area where more yolk is concentrated, more yolk is present, is called the vegetal pole in case of the blastula.